Good morning and welcome to the Center for Iron and Heme Disorders virtual seminar series. I'm Diane Ward and I am one of the enrichment coordinators for the Utah Center of Excellence in Hematology. And we're really happy to start up our virtual seminar series again this year. And our speaker today is Francesca Vinchi, who comes to us from the New York Blood Center. I don't want to take too much time, but I want to remind you, and I don't have it up here, but if you go visit the Centers of Excellence in Hematology website, CCEH.io, we have a call for new grant proposals due the 15th of this month, but certainly you should go in there and read about that in case you are interested in like a pilot and feasibility project. Today's talk is on macrophage metabolic rewiring, and you can see the title here from Francesca. She's asked us to hold questions to the end, but if you have a burning question in there that is needed to be asked, you can populate it in the chat and I will invite you to ask, or I will interrupt to ask the question. And so then I would ask individuals, you're welcome to keep your video on, but please mute yourself so that we don't have interruptions during the presentation. And thank you, Francesca, for presenting today.
So hello, everyone. Thanks, Diane, for the introduction. So today I would like to talk to you about a project that we have recently published in the lab a few months ago in Blood, and is the result of a collaboration we had with CIHD on where we, let's say, worked together, especially on aspects related to metabolism in macrophages. These, let's say, macrophages are a cell type I'm working in the lab historically since many years. And basically, I don't think to this audience I need extensively to introduce why they are relevant for iron homeostasis. So we know that macrophages essentially are capable of handling a lot of iron and acquire iron in the form of both heme and the hemoglobin, as well as transferring bound iron or free NTBI. And they handle this iron and render available again for the novel erythropoiesis. But what we were more interested in regard to this cell type is better understand the intersection between iron homeostasis in macrophages and how iron levels could alter the inflammatory or anti-inflammatory response with these cells. Probably some of you is familiar with the concept that macrophages have been in the last decade describe and define according to a dichotomous nomenclature, where on one side we have inflammatory M1 cells and on the other M2 anti-inflammatory cells. This seems a little bit strict and derives mostly from in vitro experiment. I rather prefer to think about and to use the term macrophage plasticity, because in vivo indeed we recognize let's say a kind of continuum of cells that shift from one polarization status to the other. And so the definition is much less strict than what we expect. However, still this is functional to one extent to understand whether macrophages produce rather pro-inflammatory cytokine and exert bactericidal activity according to what is described as the M1 cell, and on the other the M2 cell, which is rather anti-inflammatory, is more involved in tissue repair function. So for a long time, we have been interested to understand whether the exposure of the macrophage to iron in different sources alter the response of the macrophage. And these data are published, and to make a long story short, what we have found is that both heme and iron induce a pro-inflammatory skewing of the macrophage by activating the TLR4 signaling pathway and inducing ROS production. And so inhibiting this mechanism basically prevent this type of switching, and as well as using also chelator or heme scavenger just by basically preventing iron and heme accumulation in macrophages stop this skewing. And what I want to stress is that this mechanism is to some extent very much physiological. In fact, in health condition, we might think that upon a small tissue damage or trauma, cells release some heme and iron, which goes on the macrophage to activate and induce cytokine release, which have the role to recruit and activate other immune cells, to in turn reestablish homeostasis and promote tissue repair. So this is a very physiologic mechanism. However, you might think that on the other side, upon disease condition, when there is a status of chronic heme and iron overload, the macrophage is constantly stimulated and therefore contribute to inflammation. And we believe this might have relevance in pathologic condition. And our focus, I mean, I work extensively on a plethora of disease in this regard, and our focus today will be predominantly sickle cell disease. In sickle cell disease, we know that there is a condition of heme overload with saturation of the heme scavenger. And a few years ago, we have described that macrophages in the liver show a pro-inflammatory status, express high cytokines. And to make a long story short, we have described that this mechanism contributes to sterile inflammation and promote fibrosis in the liver of the mice, whereas scavenging of heme by hemopexin prevent this mechanism. We believe this is also relevant for patients with sickle cell disease. In fact, we found in patients, for example, with hemolysis such as efferocytosis or also sickle cell disease, an elevation of inflammatory cytokines that are predominantly produced by macrophages. And they correlate with the heme and iron status, really telling that the way the macrophage is activated by heme and iron contribute to the underlying chronic sterile inflammation that these patients have. So this is what we have put together a few years ago, describing a pathway via which iron and heme activated macrophage produce cytokines. This activates stellate cells and eventually cause fibrosis and immune-mediated tissue injury in sickle cell disease. And this could be prevented by scavenging heme and iron. However, what was out of the picture at that time was how heme and iron could alter the function, the effector function of macrophages. We just focus extremely on inflammation. So what I would like today to do is to guide you towards some aspects where we analyze the function of macrophages and how heme and iron alters them. And we will focus on efferocytosis, although in the original paper, if you are interested, you will find more information also about other functions. So we were interested in defining how efferocytosis was altered in macrophages eventually by heme and iron, because efferocytosis, which is essentially the ability of the macrophage to pick up apoptotic cell, might be key in the context of sickle cell disease, where hypoxia and vasocclusion could lead to the accumulation of, could lead to damage and the accumulation of apoptotic cell that needs to be cleared. So we asked how was efferocytosis in sickle mice, and we injected labeled apoptotic neutrophil, and we followed their accumulation in phagocytes. And as you can appreciate here, both resident macrophages, which are Kupffer cells in the liver, and also recruited phagocytes that are mostly monocytes and neutrophil, show a suppression in their ability to uptake apoptotic cells. You can nicely observe here in the upper end on the right side. So in this plot, you can appreciate that here in the Kupffer cells, in red are indicated effective efferocytes. They are clearly decreased in sickle mice, they almost disappear, and there is a slight improvement when hemopexin is administered in sickle mice, telling that this is somehow heme-dependent, this suppression of efferocytosis. And the same for recruited cell here in green, the efferocytes, the active efferocytes that almost disappear in sickle and are a little bit better with hemopexin treatment. So to further prove that this was independent, we just repeated this experiment at this point in mice that have been treated with heme. And very clearly, again, both resident and recruited phagocytes show a suppression of in the efferocytic ability. So the same was pretty clear in vitro. So you see heme-treated bone marrow-derived macrophages show a reduced efferocytic ability, and interestingly also the iron moiety of the heme molecule cause the same suppression of efferocytosis. So this occurs both upon heme and iron excess. So at this point, we asked what was the underlying molecular mechanism of this suppressed efferocytosis. And we firstly investigated the most obvious aspect, which was the expression of receptor involved in the uptake of apoptotic cell. And as you can appreciate, in macrophages of the liver of sickle mice, most of these efferocytosis receptors are suppressed. And the same suppression was fully recapitulated in wild-type mice that have been treated with heme. And in vitro, the same also in bone marrow-derived macrophages that have been exposed to heme. As you can appreciate, so some of the efferocytosis receptor upon exposure to apoptotic cell are even upregulated, such as CD36. But you see this modulation is totally suppressed by heme. So basically, macrophages are unable to express properly efferocytosis receptor, and this could contribute to heme-driven defective efferocytosis. We asked then what could lead to this suppression of the receptor, and we investigated two major transcription factors, which are also important metabolic regulator, and I will give you more insight into this later on. These two nuclear receptors, PGC1 alpha and PPAR gamma, control usually the expression of this receptor. And interestingly, what we found is that in macrophages exposed to heme, both PPAR gamma and PGC1 alpha are downregulated just at the transcriptional level. And what we did, and this will be useful for the experiment also, I will show you later on, we used two approaches to improve the activity or expression of this transcription factor. We used interleukin-4, which, as you can appreciate, directly increase at transcriptional level PPAR gamma, and we also use pioglitazone. Pioglitazone, it doesn't affect the transcriptional level of the of PPAR gamma, but it's a ligand for PPAR gamma. It induces the nuclear translocation of the transcription factor, and it basically activates its function. So it's another way to make PPAR gamma work better. What we investigated and what was very interesting to think about is that PGC1 alpha are clearly suppressed by heme, and therefore this might be responsible of the reduction of efferocytosis receptor and the linear suppressed efferocytosis. However, the function of this transcription factor is also importantly connected to mitochondrial metabolism. In fact, PGC and PPAR control mitochondrial biogenesis and also the expression of genes involved in the fatty acid oxidation. And doing so, basically, PGC and PPAR control the uptake of lipids and lipid membrane through the efferocytosis process, and at the same time coordinate this uptake with the ability of the cell to degrade, to catabolize properly these lipids through the fatty acid oxidation. That's why these pathways are basically coordinated and regulated by this transcription factor. So at this point, we asked, when heme suppress PGC and PPAR, can we observe also some kind of mitochondrial remodeling in these macrophages? And this is what we analyzed in heme-treated mice. We analyzed the mitochondrial compartment, and we found that the mitochondrial mass, both in resident and recruited phagocytes, is strongly suppressed by heme. And together with this, we also observe a change in the membrane potential and mitochondrial loss accumulation, which are increased by heme exposure. We did the same in vitro, and again, it was pretty evident that the mitochondrial biogenesis was reduced in treated macrophages, as suggested by the reduced mitochondrial mass. Confocal microscopy analysis indicated that while the mitochondrial number is indeed reduced, the mitochondrial area seems even to be a bit enlarged, like there is a kind of altered process of fission and fusion. Probably there is an increased fusion of the mitochondria in this condition. And again, ROS are elevated. As you can appreciate, the activation of PPAR gamma by pioglitazone or its induction by interleukin-4 correct most of these parameters. Another aspect that regards still the mitochondrial dynamics and we thought could be relevant for the efferocytosis was the observation that both the mitochondrial potential and the calcium in the mitochondria are elevated in heme-treated macrophages. Actually, continual efferocytosis, so the continual uptake of apoptotic cell, usually is allowed and facilitated by a suppression of the mitochondrial membrane potential and calcium. And this suppression is triggered by an induction of two mitochondrial proteins, which are UCP2 and ERP1. Indeed, here you can appreciate that heme suppress both UCP2 and ERP1, and indeed the mitochondrial membrane potential and calcium, instead of being decreased, are rather increased. And so we believe this is a secondary mechanism via which the continual efferocytic process is suppressed by heme. And again, especially UCP2 is corrected when PPAR gamma is activated, and the membrane potential is also corrected. So the observation that basically heme drives all these mitochondrial remodelings make us think that indeed also a full metabolic reprogramming can be driven by heme macrophages. And to check this, first of all, we evaluated the expression of genes that are involved in the fatty acid oxidation that are, as I mentioned before, controlled by PPAR and PGC1 alpha. As you can see, most of these genes are suppressed by heme and corrected by PPAR gamma activation and induction. And this makes us think that basically macrophages, when exposed to heme, lose the ability to catabolize lipids, and lipids also that derive from the membranes of internalized apoptotic cells. So we try to mimic this. We just before treated macrophages with lipid steady states in order to achieve a same accumulation of lipids in all the cells. And then we started to expose these macrophages to heme. And as you can appreciate, basically heme-treated cells lose the ability to consume oxygens in response to lipids. So they don't degrade lipids anymore. And in fact, their ability to produce ATP in response to lipid degradation is completely abrogated by heme. So the lack of mitochondrial biogenesis and a proper increase in fatty acid oxidation, making a way that macrophages, when exposed to heme, lose the ability to degrade lipid. And this is nicely reflected by the metabolomic analysis that we have done with James at the metabolomic course. As you can see, basically, heme-treated macrophages contain much less lipid intermediates. They have high glucose, high pyruvate, and lactic acid. So when we do when we try to revert this just using hemopexin, we could see that we could increase to some extent some lipid intermediates. So really saying that the block of the lipid metabolism is caused by heme, and when we scavenge heme, we can ameliorate the situation. Of what happened in regard to efferocytosis, it is obvious that basically the lack of efferocytosis itself, it's a mechanism that basically block the lipid metabolism and leads to a rewiring of the metabolome in the macrophage. What we did here is simply to expose macrophages to apoptotic cell in presence of heme. And as I told you before, apoptotic cells are not properly accumulated when heme is present in the microenvironment. So as you can follow here, just normal macrophages that perform a good efferocytosis, as indicated in the green line, are capable to show an increased production, an increased consumption of oxygen and production of ATP in response to efferocytosis. However, heme-treated macrophages that do not perform efferocytosis do not consume oxygen and produce less ATP. And this is obviously due to the reduced availability of lipids that come into the cells because of the reduced efferocytosis, but also to the fact, as I showed you before, that together with this, the mitochondrial compartment is remodeled, is smaller and not functional, and therefore there is no lipid metabolism. Again, also upon exposure of to apoptotic cell and heme together, we found a similar metabolic rewiring to the one induced by heme alone. So a reduction in the lipid intermediate, and as you can see here, increased representation of pathway associated with glucose metabolism, glycolysis, the Warburg effect, and pentose phosphate pathway. So I want also to stress that this is not exclusive of heme, but basically iron itself is capable of triggering a very similar glycolysis rewiring of the macrophage. So we can, at this point, we asked whether we can revert and change all these efferocytic process and the metabolic rewiring. And what we used again was the strategy to induce PPAR gamma activity and expression via pioglitazone and interleukin-4. Here you can see the example of one efferocytosis receptor, CD36, which is corrected by pioglitazone and interleukin-4, and efferocytosis is improved in presence of heme when pioglitazone and interleukin-4 are present in the medium. Obviously, efferocytosis is also improved by heme scavenging through hemopexin, and also to some extent by inhibition of TLR4 signaling pathway, indicating that this signaling might be also involved in efferocytosis suppression. And again, when macrophages exposed to pioglitazone in presence of heme are capable to perform a better efferocytosis, again, all the pathway related to lipid metabolism are increased, are activated, especially the oxidation of fatty acid, as you can see. So how we can put all this together, because I give you a lot of information, but I believe that we can summarize this finding thinking that heme basically induce a functional reprogramming of macrophages, doing two things in a very much coordinated manner. It basically triggered the coordinated suppression of efferocytosis on one side because it reduced the expression of efferocytosis receptor, and on the other, it suppressed the mitochondrial compartment and the fatty acid oxidation, which is a kind of metabolic adaptation. In fact, if the cells doesn't acquire any more lipids through efferocytosis, it doesn't need such an expanded mitochondrial compartment to perform the degradation. So the two things basically happen in a coordinated manner. How these mechanisms intersect with the inflammatory response of macrophages? Importantly, we have to think about efferocytosis as a anti-inflammatory mechanism. So the uptake of apoptotic cells through the degradation of the lipids that derive from the cells induce anti-inflammatory mechanism. As you can see, in fact, macrophages exposed to apoptotic cell start to express increased level of anti-inflammatory cytokines such as interleukin-4 and 10. However, this is blunted by heme, and heme still maintains, also in presence of apoptotic cells, very much elevated pro-inflammatory cytokines. So basically, the cells keep maintaining a pro-inflammatory phenotype and fails to rewire towards an anti-inflammatory phenotype, as it should normally happen when the efferocytosis is good, is very effective. And only the correction, the improvement of efferocytosis through interleukin-4 and pioglitazone restore the expression of anti-inflammatory cytokines and reduce the expression of pro-inflammatory cytokines. So we can put all of this together. Basically, heme, by suppressing PGC and PPAR, reduce efferocytosis, limit mitochondrial metabolism. And on one side, therefore, it reduces the ability of the macrophage to exert repair and remodeling function and to scavenge that cell. It turns the cells in a highly glycolytic cell, so it rewires the metabolome through glycolysis, and glycolysis is thought to be very important indeed for cytokine, for inflammatory cytokine expression that, as I showed you before, remain elevated while anti-inflammatory cytokines are suppressed. And others recently have also described that this mechanism, this metabolic rewiring, especially the activation of the pentose phosphate pathway that we also observed, is important to provide to the macrophage the NADPH intermediate that sustain hemoxygenase activity in a way to ensure a proper heme detoxification. So it's a coordination of mechanisms that change the function of the cells, switch the cells towards a very pro-inflammatory cells, and sustain heme degradation. So how this is relevant for sickle cell disease and for the pathology? So we went back to the sickle mice and we compared them with control mice. We injected apoptotic cell in these mice and we follow how in macrophages PPAR gamma and the mitochondrial compartment are altered. And as you can see, PPAR gamma is induced by apoptotic cell in normal mice to promote efferocytosis and mitochondrial metabolism. And indeed, when we look at the mitochondrial mass and therefore the biogenesis of the mitochondria is increased to stimulate efferocytosis and the degradation of the dead cells. Together with this, the membrane potential and the calcium are decreased. I mentioned a few slides ago that the decrease in mitochondrial in the membrane potential and in the calcium allow continual efferocytosis, so it should be so. And the ROS are also reduced. However, you can clearly appreciate that sickle mice fail to modulate any of these mechanisms. So eventually the elevated heme level that are present in sickle mice suppress PPAR gamma induction and remodel completely the left unchanged, let's say, the mitochondrial compartment doesn't allow the mitochondrial compartment to expand and to support continual efferocytosis. Together with this, also the gene of the fatty acid oxidation are increased in mice that can perform proper efferocytosis in control mice, but they are not upregulated in sickle mice. And as a consequence, macrophages produce ATP when they can perform a proper efferocytosis, as you can see, but sickle mice that do not perform proper efferocytosis don't produce ATP and don't in response to apoptotic cell. So again, also in sickle cell disease, very simply, macrophages from the liver show as well a very glycolytic metabolome. So there is a tendency to have reduced fatty acid oxidation, increase lactate and glucose, and in the enrichment analysis, we find again that the Warburg effect is prominent in these macrophages. When we provide to the macrophage apoptotic cell in normal condition, as you can see, a normal macrophage respond by activating all the pathway of the fatty acid beta oxidation. However, sickle mice are incapable of doing so. As you can see, still all the lipid intermediates remain very low. And in sickle cell disease, the macrophage, even when exposed to apoptotic cell, remain very highly glycolytic and is not rewired towards a lipid metabolism as it should be. So what is the implication of all these mechanisms, and are there pathophysiological consequences attached to the fact that heme drives this defective efferocytosis? We believe that such a mechanism of defective efferocytosis could aggravate tissue damage in sickle cell disease, and to explore this we couldn't use sickle mice because obviously you expect more damage in sickle mice because you have vaso-occlusion that in normal mice you don't have. So we used an artificial assay, and we induced apoptotic liver injury injecting thiocetamide in mice. And basically, as you can see here, we have 3 group: mice injected only with thiocetamide, mice injected with thiocetamide together with heme, or together with heme plus pioglitazone. And we analyze efferocytosis that is reduced by heme and slightly improved by pioglitazone, both in resident and recruited phagocytes. And this is associated with the suppression of the efferocytosis receptor, obviously, by heme and is ameliorated by pioglitazone as we expect. So after checking that indeed heme in this model change also efferocytosis as we expected, we analyzed tissue damage. So how many apoptotic hepatocytes accumulate and how the tissue damage is resolved. As you can see, the number of annexin 5 positive or caspase-3 positive hepatocytes, as well as the hepatic damage indicated by the measurement of AST in the mice across the 3 groups is very similar 1 day after the injury. However, after 4 days from the injury, the damage is almost completely resolved in mice treated only with tirocetamide. However, it persists in mice that are also treated with heme and partially improved, partially rescued by the addition of pioglitazone, really indicating that heme-driven defective efferocytosis leads to the accumulation of these apoptotic hepatocytes that eventually turns into necrotic cells, and they are not properly cleared in the microenvironment. And you can appreciate how the damage persists in presence of heme excess here in the histology very clearly. So what is the additional consequence of this? Not only you have an aggravated damage that keep accumulating without being properly resolved, but we think that this is also triggering some kind of autoimmunity. In fact, mice exposed to heme and treated with tirocetamide show an increased level of anti-nuclear antibody, suggesting that the cells, the apoptotic cells that remain in the microenvironment stimulate an autoimmune response of the immune system, and therefore autoimmunity could be the outcome. And I believe this might be relevant in sickle cell disease because patients with sickle cell disease that nowadays live longer, have a longer life expectancy, are more and more diagnosed nowadays with autoimmune disease. And this might be a mechanism contributing to this type of autoimmunity. So at this point, indeed, to confirm also that in sickle, this accumulation of tissue damage could be rescued by removing some heme. We also checked into sickle mice treated with hemopexin. As you can see, with hemopexin, we can alleviate the number of apoptotic and necrotic hepatocytes that accumulate. And when we analyze just by histology, the necrotic, apoptotic, and necrotic areas, they are decreased by hemopexin. So at this point, we wonder whether we could indeed properly rewire the macrophage, maybe rewire the macrophage from a metabolic point of view in order to trigger the macrophage towards a more anti-inflammatory phenotype with an increased efferocytic function in order to limit tissue damage in sickle cell disease. And we did this in sickle mice, treating them with interleukin-4. Basically, what interleukin-4 is doing is capable to improve the expression of some of the efferocytosis receptor. It improves, indeed, efferocytosis in hepatic macrophages. These macrophages start to produce more ATP in response to the catabolism of efferocytized cells. And moreover, these macrophages also turn in more anti-inflammatory cells. They show a suppression in response to interleukin-4 of a pro-inflammatory cytokine as TNF-alpha and interferon gamma. And overall, also interleukin-4 reduces the accumulation of apoptotic cell in the liver and reduces marker of damage. So last that we sought to evaluate, because we had the availability of some patient for some sample from sickle patient, is if we could observe this also in presence of plasma sample of sickle patients. We basically perform some phagocytic and efferocytic assay on macrophages, usually in the plasma of sickle patient. And as you can see, putting side by side with heme exposure, you can see that the efferocytic ability of macrophages is reduced by the plasma of sickle patient to a similar extent as heme alone. And this indeed has an inverse correlation with the heme amount that is in the plasma of these patients, and instead a direct correlation with hemopexin level. So the more is the heme, more reduced is the efferocytosis, and the highest is the hemopexin, more protected are these macrophages, and better is their efferocytosis in these patients. And at this point, we just rescue these defective efferocytosis induced by the heme contained in the plasma of sickle patients by adding aptoglobin and hemopexin in the plasma just to buffer all the sources of free heme, or by adding interleukin-4. And as you can see, both the two approaches improve the efferocytosis. And when we analyze the plasma of each single patient, you can appreciate that for most of the sample of the patients, we find in presence of aptoglobin and hemopexin as well as interleukin-4 an increment in the efferocytic ability. So overall, putting all this picture together, while earlier we have identified a mechanism via which heme and iron induces switching, a pro-inflammatory switching of the macrophage, which per se cause fibrosis in the liver via hepatic stellate cell activation, we believe now that there is an additional mechanism represented by the decreased ability of the macrophage to perform efferocytosis, which leads to the accumulation of secondary necrotic cell, also a poor release of anti-inflammatory cytokine. And this basically keeps a kind of loop where the inflammation is increased and the tissue damage is further and further aggravated. And we can use heme and iron scavenging not only as approach to block this mechanism, but also kind of metabolic rewiring, where we act by switching the cell towards a more lipidic metabolism that sustain an anti-inflammatory phenotype and more functional phenotype of the macrophage. And so these are my take-home message. I hope I've showed you that heme and iron basically induce a metabolic adaptation in macrophages, which is required to sustain the pro-inflammatory activation of the cell and also a functional reprogramming. Cells, when are activated by heme and iron, indeed become highly pro-inflammatory and functional defective in regard especially to efferocytosis, and this contributes to the tissue damage that we find in condition of chronic heme and iron overload, as in sickle cell disease. And we might take advantage of a manipulation of these macrophages for immune therapeutic purposes, trying to change and rewire their metabolome and their inflammatory properties, really to limit sterile inflammation and tissue damage that is associated with these diseases. And that's all. And last but not least, I really would like to thank all the people that contributed along the years to this project. My previous lab during my postdoc, Martina Muckenthaler and Manuela Torresano for sure. My lab that more recently has generated all this data on the efferocytosis story, and the sickle cell program that we have at the New Blood Center, which is pretty extensive. And importantly, also James and the metabolomic core that was instrumental, very important to generate all this complex picture of how macrophages work. And thanks to you for the attention, and I'm happy to take questions.
Great. Thank you so much for your presentation. We have one question in the chat. Mark, do you want to unmute yourself and ask?
Sure. Great talk, Francesca. I always just kind of sit back and absorb and try to learn as much as I can.
That's very complicated.
It's very complicated. I was curious if you tried any TLR agonists or antagonists in the presence of exogenous heme and measured PGC1 alpha and PPAR activity.
Actually, I have never, but the only, I didn't measure exactly PGC or PPAR, and you will find this piece of information in the paper. I believe that the only things we had chance to do is to measure, because we extrapolated this also from all the experiment, we measure the accumulation of tissue damage in TLR4 knockout mice. And indeed, even when they are treated with heme, they accumulate less necrotic and apoptotic cells. So they kind of probably perform better efferocytosis because the TLR4 signaling pathway is not activated and the cells remain more. So this is an indirect indication of what you are mentioning. That's the only thing we have done.
Thank you.
Other questions? Actually, I'm going to ask a question. So you're feeding the cells heme, or you're feeding the mice heme. Do you notice a difference if it's an intracellular sort of signaling piece that's causing less efferocytosis? Or I'm trying to figure out where the heme is doing its signaling process that shuts down, the ability to.
So I believe so. We didn't link the. You are asking basically the signaling pathway that leads probably to PGC and PPAR suppression, if I understood well. And we didn't, let's say, analyze the whole pathway. But what is reported in literature, and I believe it might be a hint also because we found that this seems to be partially dependent on TLR4, is that TLR4 is eventually activating NF-kappa-B. NF-kappa-B downstream is also responsible for the suppression of PPAR gamma and PGC. And this is exactly, it makes sense because NF-kappa-B triggered inflammation and it shut down and triggered glycolysis, whereas PPAR and PGC are connected to a lipid metabolism. So it just makes sense. I never, let's say, dig further into this. Probably the most interesting question is, how about iron exactly do this? Because iron is not expected to activate exactly the TLR4 signaling pathway. Probably it acts only via ROS. So I think that iron instead is via ROS eventually activating NF-kappa-B and leading to this pathway. But we didn't dissect the whole in details. We tried to hang around with the data that are available in the literature, but I think this was going beyond, But definitely is an interesting question, and maybe it's not only about NF-kappa-B.
Thank you. Other questions?
Francesca, that's a great talk.
Thanks.
So I think I might miss it, but I don't remember. Did you show that the decreased efferocytosis or phagocytosis of the SCD macrophage, is that due to the hemolysis, or like in vitro when you do cell culture, just compared to the control, the MDM, the SCD macrophage, even without the presence of extra heme, it also still showed decreased phagocytosis capacity?
No, obviously I'm comparing a control condition with the condition where I add extra heme, right? So basically it's the heme and the hemolysis that directly suppress the efferocytosis. And the demonstration comes from the fact that if you use hemopexin to scavenge, you improve the efferocytosis. I don't know if I really got what you asked me, or
I just want to know, like, is that just intrinsically the sickle cell disease macrophage has a decrease?
Intrinsically. Well, yes, in the sense that the experiment we've done in sickle mice is just injecting apoptotic cells. So we didn't inject additional heme. So if you ask at steady state, sickle mice have decreased efferocytosis, the answer is yes, because we use them at steady state, injected apoptotic cell and follow their uptake. You don't need to inject extra heme to have a suppressed efferocytosis in sickle cell disease. It's already a steady state lower. So I think that the amount of hemolysis they have already a steady state and the amount of free they have around is already sufficient to reduce the efferocytosis. Yeah.
Okay, thank you.
I probably, if you further inject him, maybe you abolish completely everything. But I don't like so much to inject further heme where there is a lot already around. It's not adding a lot of information to me. Plus, when you inject to heme in sickle mice, they really start to die. So you need to be careful.
Great.
Can I ask a question?
Do it.
That was a really beautiful talk. I learned a lot, and I'm sure I didn't capture everything, but treating sickle cell disease patients with IL-4 is really provocative. And I was wondering if you had begun to examine how systemic treatment with IL-4 would affect the entire immune response in sickle cell disease, which is really complex.
No, I never did in detail. Probably this is an interesting question because, I mean, interleukin-4 is anti-inflammatory per se, but I think the most intriguing person in what we are learning how it's changing in a complex manner all the metabolic features, and might be doing this not exclusively macrophages, but in many other cell types. Right. So definitely now I'm maybe picking up your suggestion that is very nice to broaden the horizon and try to explore more beyond macrophages, I think, and look at the immune response. The other thing we didn't do is to look also at autoimmunity, that I think would be another interesting aspect. That's a piece of information that to me, it says how much relevant could be this process for sickle patients if they develop autoimmunity to this extent. Maybe definitely this could be, I never thought about this in this direction, but I'm happy that you brought it up. Might be an interesting aspect.
It's super cool. Thank you. Great talk.
Other questions?
I have a question.
Go for it.
Hello. Yeah.
So
that's really interesting and fascinating talk. Thank you. And you mentioned that that's regulation. It's of heme and iron is going through ROS probably. So and treatment with IL-4 would induce also NRF2 and produce antioxidant as well. And so can you speculate more on the role of NRF2 in efferocytosis, and especially in the with heme loading? Do you have enough or not have enough, and why?
I mean, I can't really give you an insight on this in the sense that we never look at this specifically and we never analyze this in details. It's also very complicated because I think all the pathways of NRF2 and NF-κB are to some extent activated in these macrophages that are exposed to heme, because you kind of try to buffer also and increase the antioxidant response. Probably the question we should answer is to what extent we are overriding the antioxidant response. So even if it is activated, you may be override to a certain level where you cannot prevent the activation of all these mechanisms. Specifically, I don't have exactly a clue. I mean, in regard to the question that you made, what is the exact link between NRF2 and efferocytosis? I am not extremely knowledgeable in that. I should dig a little bit. There might be, but I don't know. I mean, I can't tell you more than that because I don't really know.
Yeah. Have you tried antioxidant treatment? Does it help?
What I have to say, which is interesting, at least this in regard to him, when we use antioxidant to rescue efferocytosis, is not rescue very well. ROS are not sufficient to correct everything. This is probably because with heme stimulation, the TLR4 signaling pathway has a major role in suppressing PGC and PPAR. So maybe ROS are not so essential for the suppression of PGC and PPAR. Regarding iron, this is another thing, and I don't have still the full picture how iron, or if it is produced even more ROS, or if there is an additional mechanism, or if just the NF-κB activation. I don't know in regard to iron. I believe that for him, the TLR4 pathway probably in this case is more important in suppressing efferocytosis via PGC and PPAR suppression rather than ROS itself. With the N-acetylcysteine, we don't have the same rescue. It's also true that I'm not a fan of antioxidants because sometimes they work as a pro-oxidant, sometimes as antioxidant because they donate and pick up things randomly. Let's say I only tried also with an acetylcysteine and never with another antioxidant, which would be, or changing a lot the dosages. But while with N-acetylcysteine, we reduce the inflammation a bit, we don't improve to the same extent efferocytosis, so the mechanism is probably also to some extent independent. That's the information I have so far.
Great, thank you. We have time for one more question if somebody has a burning question they want to ask.
So, Francesca, this is Iqbal.
Ah, Iqbal.
I got a short question for you. So in your final model, all of the effects of heme is mediated purely by TLR4? Or are there other pathways by which heme actually remodels macrophages?
Again, I can't exclude because we didn't prove that PGC and PPAR downstream are regulated exclusively by TLR4. Maybe something else is coming into the game. I can't say. I just see that with TLR4 inhibition, I see some rescue. I don't know exactly the link with PGC, PPAR, and TLR4. We didn't dissect that part. There might be.
Can you speculate? How does that effect happen? I mean, that would indicate that because it's TLR stimulation, you would think that heme is purely mediating its effect externally, right? And doesn't go inside, right?
Yes. And so far, maybe it could be. But I believe that if there is something that is also ROS associated, is because it goes internally. And so that maybe there is both the mechanism contribute, I think. Maybe also, I mean, of course, you have ROS, you have TLR for activation on the membrane, and both of the two matters. How much one matter more than the other over the different mechanisms, like NF-κB, like PGC and PPAR, I can't say. But I would not believe it's just only TLR4. That's my feeling, but maybe not. And I don't think we have still the way and maybe dissected this so much to say. There are also very poor literature how PGC and PPAR are regulated, for example. Very little only on NF-κB. And therefore, what I can tell you is that, and that's maybe also interesting, is that PPAR works as a feedback mechanism because it's activated by lipid ligands. So if you initially have poor lipid ligands that are taken up, basically, also the PPAR is not activated. So it's a feedback mechanism. Obviously not depending, I mean, it doesn't answer you the question if the heme effect is only intracellular or extracellular, but it tells you that there are multiple mechanisms that are converging like in a feedback and keep maintaining the system in a weird way. Maybe you are better able to answer to me if only the extracellular or intracellular. What is your opinion? Now I'm ping-ponging back.
I've been very just in a much more collegial way. I've been very skeptical about TLR4 stimulation by heat for a long time, including the person who first described this TLR4 stimulation by heat. So I'm just curious as to, and I've asked him that question as well, is that whether the TLR stimulation by heme is purely an extracellular stimulation or whether that heme actually gets into the cell. So at that point, even he did not have the answer to how the effect of
I think that the interesting thing is that even the ROS that are produced when the heme is picked up, they can converge on the same signaling pathway.
That is the simplest explanation. Exactly. Right. I mean, yes. So, but I wanted to hear from you as to.
I mean, to discriminate this, it becomes because they go under completely same signaling pathway, I believe so. But that's why if you also saw my rescue with the TLR4 inhibitor for efferocytosis, is not a great rescue.
Okay.
It's not outstanding. But, you know, you can never compare because you never know. Maybe I put not enough, you know, I don't like to be absolute in saying something when, you know, maybe the inhibitor was not enough. Maybe you never know. You know what I mean?
Just one follow-up question, then how do you know it's heme and not heme bound to globin?
What do you mean?
You know, that
I never did an experiment using free hemoglobin. You might achieve a similar mechanism. I don't have a clue.
okay.
I mean, probably if you induce a lot of ROS using hemoglobin, you might achieve a similar. I never did this experiment.
Okay. Thanks. Very good. Thank you.
Okay. Anyone else? If not, we'll wind up the end of the presentation and want to thank everybody for their attendance. And thank you, Francesca, for your wonderful presentation. Gives us a lot of food for thought. Yeah.
No, actually, thanks for the very nice discussion. It was really, really nice.
That's the purpose, not just to hear what you're doing, but also to, share ideas and establish collaborations. As a final comment, I want to remind you that the Centers of Excellence in Hematology has a pilot and feasibility call. If you go to the CCEH.io website, there's a call date of September 15th, which is coming up, but they're short applications and it's up to $75,000 in funding. So be sure and check that out and get your application in. Also, there is available type A funding, which is core credits. If you're going to do some use of a core, either at the CIHD or at some other core, you can apply for some funding that banks money at that core for you. So please check those out, and thank you for your attendance, and we'll see you next month in October, where we'll hear from Yatrik Shah from the University of Michigan. And so hope to see you all there again. And thanks again, Francesca.
Thanks to you. Thanks, Diane. Thanks to everyone.
All right. Take care, everybody.
Have a good day. Bye.